Hi students, so in this short recording, we will be looking at example of question related to the topic osmosis. So here is example of a question, and this question is based on the images here, figure 1 and figure 2. There will be sub-questions in the following slides. Let's look at the STEM first. Figure 1 showing a scanning electron micrograph image of red blood cells. Figure 2 shows the SEM or scanning electron micrograph of the red blood cells when suspended in solution A and in solution B respectively. Solution B is the solution used for the preservation of red blood cells. Okay, now let's look at the first question. State the tonicity of solution B and explain the rationale for using the solution for preservation. So 3 marks is allocated for this question. The breakdown of the mark will be 1 mark when you state the tonicity and 2 marks for the explanation. Now let's recall the image of red blood cells in solution B. Alright, at this point you can refer to the question slide as well. So here's the image. Okay, when you look at the image, alright, the red blood cells morphology is somewhat very similar to the figure 1, which is the cells without any solution or treatment. Alright, you can see the shape of the red blood cell here biconcave, and none of the cells appears shrunken or swollen. Okay, alright, so also the other clue here, the solution B is used for preservation. So that tells us that most likely this solution will keep or maintain the integrity of the red blood cell here. So the correct answer for the tonicity will be isotonic. When the solution is isotonic, it has same osmolarity as the cell cytoplasm. So if you mention isotonic, or if you mention that it has the same osmolarity as the cytoplasm, we can give you 1 mark. Alright, so what is the rationale why we use isotonic solution? That is because we want to prevent the movement of water due to osmosis. So therefore we can avoid cell swelling or the cell disruption. So this carries 2 marks. Here's a feedback for you to refer as well. Okay, next question. State the observation of red blood cells suspended in solution A. Explain the movement of water molecules. So here, this question carries 2 marks. So 1 mark will be for stating the observation and another mark for the explanation. Now, let's look at the image of red blood cell in solution A. So here is the image. So you can compare this with figure 1 or the image of the red blood cell under the normal condition, alright, or even in solution B, which is the solution we are using for preservation. Okay, so by comparing this image, you would see that those cells in solution A appears shrunken, and you can see there are spikes around the cell. So let's look at the answer. Okay, so the observation just merely stating what you see. So if you answer the cells are shrunken or have spikes, we can give you 1 mark. Okay, so why they are shrunken? Because the water molecules are moving out of the cells into the solution since there is less water concentration outside of the cell. So here's the feedback, okay? So from the feedback also, we would know that the solution most likely is hypertonic. Yeah, so because it is hypertonic, it has got more solutes in the solution and less water concentration. So water inside of the cell is moving out. So our cells here are shrinking. Right, so for the next question, state 2 examples of electrolytes that can be used in the preparation of solution B. This carries 1 mark. Explain what happens to the red blood cell if the concentration of the electrolytes is reduced in solution B. Okay, so total 3 marks. Okay, now let's look at the first part. It says that state the example. Now recall from the first question, we have established that solution B is an isotonic solution. Alright, that means it has same osmolarity as the cell's cytoplasm. Okay, let's look at the image here. So this is the image where we can see the shape of the red blood cell is preserved. Yeah, so what are the example of electrolytes that we can use? So think about electrolytes that you can find in the extracellular fluid. Okay, so mainly these are sodium. Okay, so sodium, it maintains the extracellular fluid's osmolality, and it is also needed in the resting membrane potential, so it works closely with potassium. All right, apart from that, you also have calcium ions, okay, that helps with the release of neurotransmitter from the neurons. Okay, you also have got phosphate here. All right, so if you think about the normal saline, okay, in the hospital, they contain electrolytes such as sodium and chloride. Yeah, so we can use this type of electrolytes as a preservation solution of red blood cell. Okay, so here you can see the allocation of the mark. For any of the electrolytes that you mention, you get 0.5, so you have to mention 2 examples to get 1 mark. Okay, so in case you happen to reduce the concentration of electrolytes in the solution, what will happen? So when you reduce the concentration of electrolytes, there will be less solutes in solution B. Therefore, now this solution becomes hypotonic. Yeah? When it's hypotonic, now water molecules will be moving from outside of the cell towards inside. Yeah? So the cells now will be swollen, because the water molecules from the solution or from outside is moving inside the cells. Okay, right? So you can see that it will start to swell. Okay, so this contributes to 2 marks. All right, so here is some explanation of feedback. Right, so the final question is state the process of membrane transport observed in the example shown in figure 2. State 1 clinical application using the same principle in the medical field. Alright, so you can see that this question carries 2 marks. So one for stating the name of the process, which is referring to the membrane transport mechanism, and another mark is for stating the clinical application. Let's look at the answer. Okay, so of course this is osmosis. Okay, we are looking at the movement of water molecules through a semi-permeable membrane. Yeah, So osmosis is the correct answer, and this is worth 1 mark. Medical application, so I think if you have gone through our practical, you would know that some of the application include dialysis, intravenous infused fluids, blood storage. So any other relevant example, we will award 1 mark. All right, so I hope the session or this recording is useful for you to see how the content of the practical can be tested in your exam, and also how you can approach an example of question which has got images in them. All right, thank you very much.